Welcome to my class, Dr. Haldar online class. Today we will be discussing on a very important topic in biochemistry, that is glycolysis. Before to going into the topic, I need to tell the importance of this glycolysis. Glycolysis happens almost every cells in our body, and mainly the enzymes of the glycolysis are present with the cytosomal fractions of the cells. Usually happens in the presence of oxygen, that is called aerobic glycolysis, or in the absence of oxygen, that is called anaerobic glycolysis. So we'll be seeing the detailed phenomena, or we will be trying to understand the mechanism, metabolism which are associated with glycolysis. So let's start. Here we can see the definition of glycolysis. It is written as glycolysis is defined as the sequence of reactions that converts glucose into pyruvate or lactate along with the production of the ATP. That is most important. Why glycolysis? Because glycolysis, our body needs glycolysis to happen so that ATP can be generated, and that ATP will be utilized by the numerous cells of our body. Fine. And as I said, that glycolysis can be occurred in the presence of oxygen, at the same time in the absence of oxygen. If it is in the presence of oxygen, then it is called aerobic glycolysis, and you can see from the definition that once glucose is converted through glycolysis, it will either produce pyruvate or it will produce lactate. So in the presence of oxygen, during glycolysis, glucose will be converted into pyruvate, whereas in the absence of oxygen, glucose will be converted into lactate. Sorry, there is some technical error in between. Okay, hope we have come back nicely. So, what are the major features? Major features we have already discussed, that it occurs almost in all the cells of the body. Thereafter, the end product of aerobic as well as anaerobic conditions both are different, and therefore, another important thing is that glycolysis is very essential for the brain, which really depends on glucose for energy, and the glucose in the brain has to undergo glycolysis. And here we can see in anaerobic condition, glycolysis, uh, what happens in glycolysis means glucose will convert into the lactate in the absence of glycolysis. And the reversal of glycolysis, we know it very well from our 8th standard, that it is called as gluconeogenesis. Now, this is the entire reactions or entire metabolism which is happening in or during glycolysis. If we subdivide it, this entire metabolism, then we can see glycolysis is happening into three different phases. First phase, here we have highlighted with yellow color, that is called as energy investment. Here, energy will be invested to continue or to carry on these processes. Second, this is called splitting phase, where the product will be splitted into two different, or the substrate will be converted into two different product. That is called the splitting phases. And finally, these are the steps which we can mark with the red or highlighted with the red, is known as the energy generation. Here you can see in the very right side extreme down, we have mentioned the different phases. First one is energy investment, that here it is highlighted with the yellow, and second was the splitting, here it is highlighted with the green, and the final one is the energy generation, which is highlighted with the red in color for you to understand it clearly. Now see phase by phase. First one is the energy investment phase, where you can see that glucose in the presence of the enzyme hexokinase is getting converted into glucose 6 phosphate. This is the first most important reaction or metabolism reaction in glycolysis, where we can see glucose has converted into glucose 6 phosphate. We know that glucose is nothing but a 6 carbon monosugars, Six carbon monosucres means if we calculate or if we count from this end, then one, two, three, four, five, six. This carbon is the sixth number of carbon. Now you see, in the presence of the enzyme hexokinase over here, what is happening? Glucose is getting converted into one product which is called as glucose 6 phosphate, and we can see that the one phosphate is added to the carbon number 6. That's why it is called as glucose 6 phosphate. Next, what is happening in the presence of the enzyme phosphohexose isomerase? Glucose 6 phosphate is getting converted into fructose 6 phosphate. Why the enzyme is so called name as phosphohexose isomerase means this glucose 6 phosphate and fructose 6 phosphate, they both are the isomers, and we know isomers. What are the isomers? We have studied in our plus 2 levels. What are the isomers? So because of the involvement of this enzyme isomerase, so glucose 6 phosphate is getting converted into the another isomer that is called fructose 6 phosphate. Then the third step is, now in another enzyme that is the phosphofructokinase, fructose 6 phosphate is getting converted into fructose 1 6 bisphosphate. Okay, now here we can see the steps what are happening here in the first and third step. It is happening with the aid of ATP, means these reactions can be carried forward only with the help of ATP. That's why ATP is being invested over here in these steps. That's why these steps are coming under energy investment phases. Fine. Okay, let's move on. Then fructose 1,6 bisphosphate. This is the second phase of the glycolysis, that is called splitting phases. So splitting phases means the substrate will get converted or will be bifurcated into two different product or phases. So here, what is happening? The product which was produced in the energy investment phase will be splitted in the presence of the enzyme aldolase in the splitting phase. And what it will form? It will form two different product, that is dihydro acetone phosphate as well as glyceraldehyde 3 phosphate. Now you see all these means still here, starting from the glucose, all they were the 6 carbon atom. Fine, 6 carbon molecules. Now here we can see those 6 carbon molecule, that is the fructose 1,6 bisphosphate, is getting or bifurcated into two different molecule which are three carbon molecules individually. Means the dihydroxyacetone phosphate is a three carbon molecule, as well as glyceraldehyde 3 phosphate is a three carbon molecule. Here you can see 1 2 3. Here you can see 1 2 3. So both are bifurcated into 3 carbon atom based molecule. Fine. Why it is dihydroxyacetone phosphate? Because here acetone group is there as well as phosphate group is there. That's why it is called dihydroxyacetone phosphate. Initially there was a hydroxyl group here at the one position, then another hydroxyl group was here, then one hydroxyl group is replaced by the phosphate group. That's why it is called as dihydroxyacetone phosphate. Now here why it is called glyceraldehyde 3 phosphate? Because the glyceraldehyde is nothing but one of the aldehyde, and we know the functional group of aldehyde is CHO. That's why CHO is there, and 3 phosphate because the third carbon, the hydroxyl group, is replaced by the phosphate molecule. And both this glyceraldehyde 3 phosphate and dihydroxyacetone phosphate has a tendency to interconvert to each other with the presence of the enzyme that is called phosphotriose isomerase. And for our own understanding or remembrance rather, we used to call digoxin acetone phosphate as a DAF and glyceraldehyde acetone 3 phosphate as a GAP. Okay, so this is all about the splitting phases. So we have completed the energy investment phases, we have completed now the splitting phases. Let's see how energy generation occurs in glycolysis. These are the very important few steps. Okay, let's see one by one. Now we have obtained glyceraldehyde 3 phosphate. Fine. So in the presence of the enzyme glyceraldehyde 3 phosphate dehydrogenate, what it is happens? Now you know what is the role of the enzyme dehydrogenase. Dehydrogenase will take the hydrogen atom from the substrate molecule and it will relieve it or it will eliminate it, which will be quite obviously accepted by some of the nucleotide. Here see what is happening. Glyceraldehyde 3 phosphate dehydrogenase has come into the picture. What it will do, it will just extract the hydrogen molecule from glyceraldehyde 3 phosphate from its substrate, and then what will be the fate of that hydrogen molecule? That hydrogen molecule, or that hydrogen ion rather, will be accepted by the another nucleotide. The nucleotide over here is NAD plus. Once it will be accepted by NAD plus, it will automatically be converted into NADH plus H plus. And what will happen for glycerol there 3 phosphate, that is a substrate, this will automatically will get oxidized into 1,3 bis phosphoglycerate. This is one of the most energy containing product in glycolysis. If I'm not wrong, then it is the most energy content compound in case of glycolysis. So at the same time, we can call this step as a substrate level phosphorylation, because here they are getting the energy, not using any ATP and all. They are to using some substrate. Let's see how it happens. 1,3 phosphoglycerate is getting converted into 3 phosphoglycerate with the presence of the enzyme phosphoglycerate kinase. Here we can see that here energy is getting generated. Here, when phosphoglycerate kinase is coming into the picture, what it will do, it will just convert 1,3 phosphoglycerate into 3 phosphoglycerate. And we can also see side reaction that ATP is getting converted into ATP. Means these are the steps where energy is getting produced. Fine. Then, one 3-phosphoglycerate is present, then in the presence of the enzyme phosphoglycerate mutage, 3-phosphoglycerate is getting converted into 2-phosphoglycerate. A slight minor mutation is occurring because of this enzyme mutation. So the phosphate which was there in the third position in case of 3-phosphoglycerate is now being transferred into carbon number 2 in case of 2-phosphoglycerate. This is a slight minor change or mutation occurs. That's why this called as a this step is being rather carried out in the presence of phosphoglycerate mutage. Fine. Now we have 2-phosphoglycerate in our hand. Then it will be converted into phosphoenol pyruvate, another energy containing compound in glycolysis. And this step will be performed, or this step will be happening in the presence of the enzyme enolase. And this is important as far as diagnostic centers are concerned. When in those centers, people go for measuring or getting their glucose level in the blood. So for the estimation, what they do, those experts, what does they do, those pathologists rather, they add some fluoride ion. Okay, so they add some fluoride ion just to give pause for the glycolysis to further occur, so that they can find out what is the level of glucose in their blood. Okay, so just to get an estimation, just to have an idea, just to get the value for the glucose level in their blood, what they do, they just mix enolase, so that glycolysis cannot proceed further. And those pathologies will be able to find out their find out the glucose concentration of the blood, or in the glucose concentration present in the blood of the patients. Fine. So we'll be knowing those things in detail later. But now let's see how glycolysis is happening further. Once phosphoenol pyruvate has formed, then in the presence of the enzyme pyruvate kinase, it will again convert it into pyruvate. So this is the end product, and pyruvate can be there, can be occur there, can be formed in two forms. One is enolic form, that is nothing but the alcoholic form, and another one is the ketonic form, that is due to the presence of the ketonic group, that is acetone. Okay, so till the formation of pyruvate from glucose is comes is regarded as the aerobic phase of glycolysis. However, even here we can see, even we will discuss it later in some slides, that in the presence of lactose dehydrogenase, pyruvate will get converted into the lactate. Okay, this step, specifically this step, pyruvate to for lactate, or formation of lactate from pyruvate, comes under, or usually happens in case of anaerobic conditions. Okay, so this is the overall basics, overall an idea on the metabolism of glycolysis. We are not going so detailed into the glycolysis. However, in our subsequent classes, we will be discussing on several different MCQs and different important questions from glycolysis, especially from the metabolism point of view. And so that our knowledge in the glycolysis will be enhanced. So thank you.